Hello, my name is Bill Longabaugh, and I'm a senior software engineer at the Institute for Systems Biology, and today I'm going to be talking about cloud resources and computing. So first off, I'll talk about cloud resources and how they fit into the NCI Cancer Research Data Commons, and then I'll give an overview of each cloud resource, which includes the Seven Bridges Genomics CGC, Broadfire Cloud, and the ISBCGC. So first of all, the diagram of the whole CRDC is provided on the left with the details showing the nodes, the data nodes on the top there, including proteomic, genomic, imaging, etc. And then below that, we show the NCI cloud resources, which is intended to be the interface through which the various users of the system will be interacting with the data. So again, the underlying principles of on-premises versus cloud computing is that instead of everybody having to make their own download and keep their own copies of the data and deal with them separately, the idea is to have a single copy of the data that everybody shares, and with cloud resources providing tool sets to work that are next to the data and work next to the data to provide a centralized resource for the entire community. So the way I prefer to think about it is that cloud resources sit above the data nodes. The data nodes are responsible for data hosting, curation, domain-specific search tools, while the cloud resources are about computation and shared workspaces, collaboration, and doing cross-omics analyses. So the cloud resources have a view to all of the various different types of data and allow the user to combine data from the various different nodes and do their analyses in the cloud. So to get to the cloud resources, you can go to the CRDC data portal, URLs at the bottom, go to the Analyze item on the top menu, and you can get links to the various cloud resources. A little bit of quick history. 2014, NCI created the cloud pilots, and they were tasked with figuring out how to do sharing of genomics cancer data in the cloud, controlling access to controlled access data, and providing ready-to-use cloud-based analytics tools next to the data. ISB, Seven Bridges, and Broad were selected, and the end of those initial 3-year cloud pilots, they became part of the CRDC and are known as cloud resources. One thing that happened was the role of hosting the genomics data was peeled off from the pilots, and that was put into the data nodes, such as the GDC and the other nodes coming online. And access to the data is, for controlled access, is handled through the Data Commons framework, which also handles indexing all the data in the CRDC. Since the resources are on the left got started, they're now, many are tied to a variety of different multiple NIH cloud initiatives, including Anvil, Biodata Catalyst, and Kids First. One thing to keep in mind is where does the data live and where is the compute happening? So some data nodes host data on only one commercial cloud platform. So for example, proteomics and integrated canine work on Amazon AWS. The imaging data commons, however, works on Google GCP, and the genomic data commons host data on both. Similarly, some cloud resources are centering on running computations on one platform, such as Broadfire Cloud and ISBCGC are on Google, where Seven Bridges CGC started off on Amazon, but now has the ability to run workflows on both. So you really want to keep this in mind. You want to run your computations on the platform where the data lives if the data sets are large, because egress costs can be very expensive. So quick comparison of the cloud resources, SBG and Broad. The user projects are within the resource itself. So you work entirely within those resources using their tool sets and their framework. ISBCGC is a little different in that user projects are standalone Google Cloud projects running on the Google Cloud platform. CGC provides resources for those user projects that they can access. And if they're using controlled access data, ISBCGC has a monitoring function to ensure the project is set up to handle the controlled access data. So pretty much the cloud resources all run in a similar fashion in that you set up a project, you select the data that you want to use either by building patient cohorts, choosing from existing cohorts, or importing cohorts that are created at a data node. You select files for the cohort and can attach private data to your project. You then build your workflow, creating either one from scratch or choosing from extensive libraries of existing workflows. You run your pipeline. You can interactively work with your results using Jupyter Notebooks and RStudio typically, and then you can share your results with your project members. So starting off with 7 Bridges Cancer Genomics Cloud. To build cohorts, they provide a data browser, which provides views across a number of CRDC data nodes to allow you to find that data and filter on it, find the data that you need, and use that graphical user interface to create your manifests of data that you want. Similarly, they have a public apps gallery, which provides a variety of different workflows that you can access and pull into your analysis. If you wish, you can build your own workflows using their Rabix Composer to do have a graphical user interface to build your workflow. And at the end of it all, you can run Jupyter Notebooks and RStudio sessions within the CGC, choosing the compute requirements that you need for your particular VM, and start those up and run those within your project on ISBCGC. So Broadfire Cloud is the next resource. Again, they have many existing workspaces to clone. You need to set up a workspace, and they provide a number of different existing workspaces that you can go in, find something nearly what you need, and clone it to your own workspace, and then customize it as you need. There's many workflows available as well on Broadfire Cloud. In addition, you can find them and pull them in from DocStore. Their Cromwell workflow engine allows you to create pipelines using either WDL or common workflow language, so you have that flexibility. They provide you a wide variety of data sets that you can browse those data sets, and it will take you into pre-built cohorts for the data that you can select. And if you click on a particular cohort, you then can get an associated workspace, which again you can clone into your own workspace to use as a basis to start your own analysis. They also have Jupyter Notebooks and RStudio that you can launch and run within Broadfire Cloud. Finally, ISBCGC, which I'm most familiar with because that's the project that I work on. So at ISBCGC, we also have the Data Exploring Cohort Builder that you can use to create the manifest of files ultimately that you want to use in your analysis. You can find the cases and samples of interest using the filters and the attributes within the cohort builder. And then you can find files associated with for that cohort, choose the types that you need, and then export it into your Google Cloud project. And I'll come back again here to saying that again with ISBCGC, the user projects are standalone Google Cloud projects that the user works in. So in your Google Cloud project, your Google Cloud console is your workspace that and your interface for working in that realm. You can use, for example, any number of Google APIs that you wish, such as the Life Sciences API, to build your own compute platform that you wish to do. So this is sort of it's closer to the machine in terms of how you can work with the Google Cloud system. We provide pipeline examples to get you started that you can look at and use to set yourself up on in your own project. We also provide Python and R notebooks that you can use to run analyses. One thing our notebooks are good at is to work with BigQuery. So ISBCGC offers Google BigQuery, which is a serverless data warehouse, highly scalable over very large amounts of data. It's a columnar database format, and it's ideal for storing tabular data. You can use SQL to query it and do joining of tables. And one thing we do is we basically combine large quantities of files that are available in the data nodes into single tables within Google BigQuery that you can use to run analysis across all of this data. We have a BigQuery search UI to allow you to find the tables that you wish. And I'll wrap up by pointing out that you can authenticate to get access control data on any of the resources. It basically takes you to the NIH authentication services you log in with your URA credentials, and from there you can access controlled access data. And I'll wrap up by saying that NCI cloud credits are available, so you should visit each cloud resource to get further details. And thank you very much for your attention.